Well, good afternoon, everybody, and welcome to today's webinar. Today, we're going to be giving you a summary of the outcomes of the Run 15 variant classification webinar, well, EQA, should I say. This is a non-promotional webinar organised and funded by AstraZeneca and MSD, and it's designed to be educational for you, to feed back to you on the outcomes of the EQA that we've just run in October of this year. Next slide, please. So the normal housekeeping rules, we've automatically muted your microphone to avoid any background noise. We are recording the webinar, as always. We will make it available to you as soon as we can after the webinar is concluded. Normally takes 10 to 14 days and will be available on the EMQN and GenQA YouTube channels. You will be notified by your EQA provider when that's available. But most importantly, these webinars are educational and we also need to learn just like you guys do. So we have a facility at the end of it to allow you to submit your questions to the panel. So please do use the Q&A. So you'll find the Q&A at the bottom of your Zoom browser to submit your questions. A big plead, please, when you do so, that you make sure that you very clearly state which variant case you're talking about. We have 6 variants to run through, so helpful if you just put down which variant it's applying to. Sometimes it's not easy to tell, and it does help massively for myself and my co-chair to triage the data coming into our speakers to do so. Next slide, please. Feedback. So we do value your feedback. We want to improve in the delivery of our activities to you. So you will receive an email after the webinar with a link to a short feedback survey. Normally takes no more than 2 minutes to complete, and it really helps us as well make sure that we're doing a good job and collate your feedback and get ideas for the next webinars, etc. So please do contribute your time to that. Gratefully received. Okay, next slide, please. Right, without further ado, I'm going to introduce my co-chair, Professor Sandy Deans, who's then going to introduce the speakers for today's webinar. Sandy, over to you.
Great, thank you, Simon. Welcome everyone to our Run 15 webinar. So as Simon mentioned, we have an expert panel with us today and we're absolutely delighted. We call them our dream team. So we are joined by Professor Kathleen Claus from Ghent in Belgium, also Miranda Durkee from Sheffield in the UK, and Professor Thomas van Overheem Hansen, who's from Copenhagen in Denmark. All 3 have been incredibly supportive and have been with us delivering these EQA runs for many, many years now. So they're well versed in classification of the BRCA and HRR gene variants, and also are happy to answer any questions at the end of this webinar, as Simon mentioned. So please do put your questions in the Q&A box and we'll hopefully be able to address them later on. So next slide, please. So these are our disclaimers. And next slide, please. So we're going to give an overview today of the BRCA1, BRCA2 and other homologous recombination repair gene variant classification assessment that was delivered during October this year, 2025. And this is now Run 15. Can you believe we've now completed 15 runs of variant classification? Next slide, please. So as always, this has been provided using the GenQA Genomic Online Individual Education Platform. The platform is available 24/7, 7 days a week on the GenQA website. There's multiple different education and competency assessment modules available for you to use, so please go in and have a look. These EQAs, this Run 15 EQA, has been run as a time compliant EQA, whereby it opens and closes and then we're able to analyze the data, and we're presenting to you that today on the webinar, and also we'll have a EQA summary report follow up for you to assess. Next slide, please. So one of the benefits of delivering large EQA runs like this is we actually get some data. We can actually find out what's happening globally in the real world. So here, as I mentioned, this Run 15, not Run 14 here, was delivered in October this year. We had over 400 individuals registering to participate, with just over 300 actually completing the assessment. What is absolutely fantastic, we do have global outreach, and we had participants from 44 different countries. And along the graph here in the bottom, you can see the spread of that global reach. Just to say, our top 3 countries of participants, the numbers are, first of all, UK, and not far behind is actually Poland. Then we're followed by the other European country, Germany. But here again, you can see the spread. We also have not just European countries representative. We've also got Thailand, Brazil and Argentina. So as I say, it goes across different continents, which is fantastic to see. Next slide, please. We can also find out which guidelines are currently being used by participants. And here, we're just from comparison, you can see on the left, we have the data from Run 15, the current one. And then we also have the data from Run 14, which was provided earlier in 2025 in the spring. I think the things to note here is, surprisingly, in this run, we've got 39% of individuals who don't actually state which guidelines they use. So if that was you and you're participating in future runs, we really would like you to identify which guidelines or combination of guidelines you have applied, because it does make it very much easier when it comes to assigning correct classifications to your submitted results, because as you'll see, some of the classifications can change according to which guidelines have been applied. What other thing to note here is, what's jumped out at me was actually there's fewer individuals using the ACMG 2015 only guidelines in this run. So that's reduced from 23% down to 16%. And I think that's something that we would encourage, because obviously there are gene-specific guidelines available for these genes that we're reviewing, and it's appropriate to use those where they're available. Next slide, please. We can also find out the background of individuals who are participating. So if we look at the specialties here, no surprising, the majority of individuals, over 50%, are actually coming from genetics or clinical genetics. And again, a considerable number of individuals have not listed their specialty. So if it is another one that has not been listed on the drop-down menu, please do let us know, maybe pop it in the chat, the Q&A for this session, and we'll be able to expand that list going forward. Same goes with the expertise of participants. Predominantly, and I think no surprises really, the majority are either experienced scientists, that's 41%, or another 12% who are recently qualified scientists. So no surprising that most of the variant classification happens with the scientific teams. Again, there's a large number, 20% of you did not submit which area of expertise that you were comfortable to sit in. So again, maybe pop that in the Q&A or the chat so we can expand the options going forward so the data set is more complete. Next slide, please. So a summary of the 6 variants that were included in Run 15. You can see here we've got the coverage between BRCA1, BRCA2 and 3 other HRR genes. We have a range of different types of variants. We have insertions, we've got deletions, exon deletions and substitutions as well. And again, we try and cover a full range of different classifications to make sure that we're assessing the whole scope of that activity. And as I mentioned earlier, that classification can change considerably depending on which guidelines you apply. Next slide, please. So this is a very high-level summary of the number of variants who are being correctly classified across the whole run, so across the 6 variants by the participants. The Run 15 data is on the graph on the left, so we've got 81 and 77, and then a further 55 participants who are classifying 4 or more variants correctly. So that's, we have got in previous runs here on the right-hand side, you can see the change. Now you'll see here highlighted in green, for Run 14, we had a much higher percentage of those given expected classifications up at the 50% mark. And if we move to the next slide, you'll see the graph changes, and this is the data from Run 14. And I think we've done a deep dive into the data, and I think the expert panel and Simon and myself do realise that we think the variants were slightly easier to classify, maybe less challenging. So we did up the ante for Run 15, and I think that's been demonstrated that even though a lot of people may be very competent in defining the classification, it really does depend the gene and it does depend the variant specifically. So I think we're all still on a learning curve. Next slide, please. Right, my great pleasure now to hand over to Thomas, who is going to start off the reviews of the 6th variant with variant number 1. Thomas.
Thanks, Sandy. So I will start with the first variant. I hope this works. Yeah, fine. So the first variant is a BRCA1 variant. It's insertion of a nucleotide in the last part of the gene. It does introduce a frame shift and a termination codon, but the termination is located after the original termination codon. So that's rather important. And it's a frame shift, and it's also located in a well-defined functional domain. If we look at databases, it's absent in Nomad version 4.1. In Cleanwar, it's reported once as likely pathogenic, and in BRCA Exchange, it's not yet reviewed. And here we look at the final classification. So if you use the VSEP Enigma guidelines, you can get the criteria PVS1 very strong, but that's the only criteria that you can get, and therefore the classification is a class 3, a VUS. In contrast, if you use the CANVIC guidelines, then you can use PVS1 very strong, but you can also use the PM2 moderate. It's because it's absent for Nomad, and I will come back to that difference a little later. So using the CANVIC guidelines, the variant is classified as a class 5 pathogenic variant. And this is the summary for this variant. You can see that the class 3 and the class 5, as expected, depending on the guidelines used, has a fair amount of answers that are correct. But you also have a large group that has classified the variant as class 4. And then you can see to the right that the guidelines used is the Enigma guidelines is the most common guidelines used, but also the CANVIC and some other guidelines. And just to look at the numbers, actually 119 participants classified this variant as likely pathogenic, 113 as pathogenic, and 79 as a VUS. So there's a lot of this variant had some difficulty for sure. If you look at the variant at the gene level, as I mentioned, it's located in the last exon. You can see the position with the red arrow, and it's you can call it an extended incorrect terminus variant. And it does not create a premature termination codon, but it creates a termination codon after the original termination codon. So you get an incorrect number of amino acids, 26 in this case, and you lose a correct number of amino acid residues, 13 in this case. And you can actually follow the PVS criteria here in the PVS1 decision tree for BRCA1. And if you can see the red box, this is a nonsense, or you can see before the red box, it's a nonsense and a frame shift variant. It's a PTC variant, but NMD is not predicted because it's located in the last exon. And it's and if it truncate or alter a region before position 1854, then you can actually make PVS 1. And that's actually the case here. As you can see, the first amino acid that is changed is on position 1851. And just to give an example that it's actually important to use the BRCA1 gene-specific guidelines in this case, because if you use the PVS decision tree suggested in 2018, then you would actually get a lower criteria as a moderate or even as strong. So again, it's important to use the gene-specific decision trees if possible. Variants like this have actually been examined functionally before, not the exact variant, but other variants that have incorrect protein C terminus. And in this article, 17 variants were investigated, and actually most of them could be considered as pathogenic using, I think, both clinical data, but also functional data. And only one of them actually did not perform as a wild type in this functional assay, and is actually now classified as a likely benign variant. But I would say that in general, these variants actually are suggested to be pathogenic. I will just come back to this. So regarding the PVS criteria, a lot of participants used this criteria, but they used it at different strength. So a lot of participants used it like PVS1, which is actually the correct use, but others used it at lower strength, with a lot of participants using the PVS1 strong. And actually 78 participants did not use PVS1 at any strength. So I mean, again, it's important to I mean, it could be very helpful to use the PVS gene-specific decision tree in this case, because then it can really help you guide you to use the correct strength of this criteria. And then we have this point with the population data. This variant is not present in NOMAD version 4.1, and therefore a lot of participants actually used PM2 supporting or stronger criteria. And if you use the CANVIC guidelines, it's actually PM2 moderate you should use, and most participants using these guidelines actually also used PM2 moderate, but still 14 participants using these guidelines used PM2 supporting. And a lot of participants using the ENIGMA guidelines used, as I said, PM2 at different strength. And if you carefully read the ENIGMA guidelines, it actually states that you should not apply the PM2 criteria if it's concerning insertions, deletion, or Dillon's variants. And we have had some discussions in our group, and yeah, we can maybe come back to that in the last part of this session. But as it is right now, you cannot use PM2 for insertions, deletion, and Dillon's variants if you use the ENIGMA guidelines, and that's the main difference in the classification here. That's why the ENIGMA guidelines only, if you use these guidelines, the variant is a VUS, and if you use the CANVIC, it's actually a class 5 pathogenic variant. A lot of participants actually also used the PM5 PTC criteria. And that's a criteria that has been used by the BRCA1 and BRCA2 VSEP as a code or as a help if you have seen a protein truncating variant in that exon before that has been determined to be pathogenic. If you then find another one, then it can actually help you get the variant that you just have found as a pathogenic variant, and not only as a likely pathogenic variant. So, and that's actually making really good sense. But you should be careful when you look at the last exon, because actually in this case, and I don't have the picture here, but if you look at the table 4, it actually is stated that if you are beyond the position 1854 in BRCA1, then you cannot use PM5 PTC strong, but if you have the protein truncation before that, then you can actually use it. So it's understandable that there's a lot of this is actually really hard if you don't look carefully in these guidelines. But actually it means that PM5 PTC cannot be used for this variant. And just another criteria that have been used by several participants, it's the PP5. I mean, it's a criteria that you have seen. The variant has been reported at databases before as pathogenic, and if you use the CANVIC or the ENIGMA guidelines, you cannot use this criteria. So it was only just to make this point for future classifications. And for the last slide for the variant 1, this is just the results by the guidelines used. So you can see a lot of participants actually used the ENIGMA guidelines. It was almost 77%, and then 84 used the CANVIC guidelines, 21 used others, and then a couple used the ATM VSEP guidelines. And of the 241 ENIGMA participants, 30% correctly classified the variant as a VUS. And of course that's due to the things that I've already stated, but as I already said, a lot did actually not use PVS1 at any strength. And strikingly, also 88 classified the variants as pathogenic using the ENIGMA guidelines, and that was actually mainly due to the use of PM1 PTC strong. And of the participants using CANVIC, 35% correctly classified the variant as pathogenic, and also some people classified it as slightly pathogenic. So for sure, that was not the most easy variant, this one. Okay, I will continue with variant 2. And variant 2 is a variant in BRCA2. It's an exon deletion, a deletion of exon 10, and there's no known functional domain in relation to exon 10 currently. If you look at the databases and look at NOMAD structural variant database, this variant is absent. And when you look at the criteria used, it's actually almost the same using the VSEP or VSEP ENIGMA or the CANVIC guidelines. You use, because it's absent, you use PM2 supporting using ENIGMA, and you use PM2 moderate using the CANVIC guidelines. And then you have some evidence, functional evidence that this deletion does not affect the activity of BRCA2, and therefore we have used the BS3 moderate in both in the VSEP ENIGMA and the CANVIC guidelines. So that ends up as a classification as a class 3 variant of VUS. And looking at the data here for this variant, actually a lot of people did classify the variant as a VUS. And again, ENIGMA was the most used guidelines, followed by CANVIC, and also some using RIGS and others. And this is just the numbers. You can see 201 participants classified this as a VUS, 44 as pathogenic, and 33 as likely pathogenic, and so forth. So there were more consensus about this variant than the former variant. And this is just a picture of the exons and the protein domains in BRCA2, and the red box show exon 10 that is deleted, and there is some phosphorylation sites, but actually there is no known functional domain in this area. It's an in-frame deletion of 371 amino acids, and that's important when you use the PVS1 decision tree. So So again, just follow the gene-specific PVS1 decision tree for BRCA2, and this is for deletions. And you can see that the first you have to consider is whether this is targeting the PALB2 or the DNA binding domains, and it's not targeting, sorry, these, and in this case it's not, and it's in frame. It removes more than 10% of the coding sequence, and exon 11 is not deleted, and that's why you end up with a PVS1 NA not applicable. So you cannot use PVS1 in this case. And again, that would actually have been different if you have used the original PVS1 decision tree from 2018. So again, it's important to use the gene-specific PVS1 decision trees. And this is the data that have examined this variant functionally. They in this study did a lot of exon deletions, and you can see the wild type activity for BRCA2 with the blue arrow, and then you can see the activity when you delete exon 10. And it's in the article they state that at least 80% activity is still present. So the effect is really low for this variant, and there's still a lot of activity left for homolog recombination. And here it's then you use the benign criteria for functional assays. That's BS3. And in the ENIGMA guidelines, I think it's only stated when you look at the guidelines as a strong evidence, but actually it has been moderated dependent on the assay and the article, how many controls are included in these assays. So it can also be used as moderate or as supporting even. And the strong evidence is mainly for calibrated assays, also including clinical data. And this group that has performed this assay in Holland, they have before examined several variants where in the Canvig functional table, they have been assigned a BS3 moderate criteria, and we have also used that for this deletion, even though it's this article is a year later than this one. There was, as you saw before, 77 participants that classified this variant as pathogenic and likely pathogenic, and that's mainly due to the use of PVS1, either as very strong or strong. And 30 participants classified this variant as likely benign or benign, and a lot of them used the functional data as benign strong. And that's really something you have to we agree that that is difficult to assign this, but sometimes there is tables to help you, and that is the case in the Canvig guidelines. PM4 was also used by some participants, and again, that's that is protein length changes due to in-frame deletion insertions, and actually that cannot be used when you look at the VSEP guidelines. So that's also something to remember. And just to look at the results by guidelines used, again, a lot of participants used the ENIGMA guidelines, followed by the Canvig guidelines, but also RIGS and others were used. And as you saw in the start, almost 70% correctly classified the variant as a VUS using the ENIGMA guidelines, and it was a bit lower amounts, almost 60% using the Canvig guidelines, and even a little bit lower using the RIGS guidelines. So I think that was variant 2. So over to you, Miranda.
Thank you, Thomas. Really interesting variants. So I'm going to do variants 3 and 4. So starting with variant 3, this is a BRCA2 variant. So it's C.6147 to 6149 del. So that's an in-frame deletion of just one amino acid. Just looking at our kind of key databases and resources, it was seen 3 times in Nomad version 2.1.1. Twice in version 3.1 and twice in version 4.1. So you might be wondering why the numbers have gone down, as most of the Nomad numbers tend to go up, but there has been a lot more QC curation, particularly in version 4. So they've obviously removed one of the alleles that was in Nomad version 2 due to low quality. The variant is listed on ClinVar 4 times as a VUS or uncertain significance, so therefore it gets 2 stars because you've got more than 3 labs in agreement, and it's not been reviewed on BRCA Exchange. So the applicable criteria is for the Enigma guidelines, it would be BP1 strong. So this code was originally in the Richards et al. Guidelines used for missense variants in genes that where primarily truncating variants cause disease. So a really good example of that, something like the APC gene. But it's kind of been repurposed in the Enigma guidelines to also include in-frame deletions outside the functional domain with no impact on splicing. In the CANVIG guidelines, we aren't quite in alignment, and we haven't yet started using that BP1 at strong. We're in discussions with the steering group about whether we adopt that. But so for CANVIG currently, there are no applicable codes, and therefore that variant would be a VUS. So having a look, and actually participants did very well for this variant. As you can see, nearly everybody got it either as class 2 or class 3 as expected. And then on the right-hand side, if we look at that, we would expect that if you're using the CANVIG guidelines, everyone would get it as a VUS, which pretty much everyone did. And if you were using the Enigma guidelines, that everybody would call it a class 2, which more have than a VUS, but they're still quite using the Enigma guidelines that have classified this as VUS. So just have a little look at the data for this variant. Decipher has a really nice browser where you can kind of really view the location of your variant with the functional domain. If you follow that kind of red arrow, it's kind of a gold line going down. You can see it's outside the key functional domains, which for BRCA2 is the DNA binding domain. If you go into the browser, you can actually go into the sequence and you can look at conservation in different animals. And I've highlighted this variant in the red square. You can see that actually it's not present in most other animals at this position, only in humans and chimpanzees. So really not kind of conserved, and the whole region is not very well conserved. We also need to look at splicing, and so you can see from the Splice AI score, the delta score is either 0 or 0.01 for all cases, so no effect on splicing. And we can use this really nice figure that's in the Enigma guidelines. It's figure 1A, and we just kind of follow this along. So we've got an in-frame, we've had a look at splicing. We've seen there's no impact on splicing. We've seen that it's not inside a functional domain. Therefore, we can apply BP1 strong. I forgot to put the screenshot of the Enigma guidelines showing that you can use BP1 strong as a standalone, but I will include that in the final scheme report. But yes, according to the Enigma guidelines, just that one code is enough for likely benign. Moving on to the CANVIG guidelines. So in CANVIG, we actually specify kind of on the pathogenic side which specific residues you can apply either PM1 or PM4 for an in-frame deletion. You can see that the 2050 for BRCA2 is not listed in that, so we know we can't apply PM4. Unfortunately, we don't quite have that kind of in the reverse in the benign side. So you can see under BP1, we haven't updated that to, as Enigma have to say, used for missense and in-frame variants. Currently, it just says missense. I think we'll be updating that really soon. The other code that we potentially could use for this is BP3, which is the in-frame deletions, insertions in a repetitive region. But actually, we've just had a look at that sequence. It wasn't very well conserved, but you wouldn't say it was a repetitive region. So you don't have any codes that we can apply. So just have a look at the results compared to the guidelines used. So nearly 75% of participants use the Enigma guidelines, and then kind of majority of the others use CAMVIG. A few saying ATM VSEP, which we presume because, you know, obviously this is for BRCA2 gene, so that VSEP guidelines would not be appropriate, but they're probably just the Enigma guidelines, and 26 other. So if we group together the Enigma and the ATM VSEP guidelines together, then 53% correctly classified it as likely benign, 2 scored it as benign, and 107, 45% as VUS. And then there's 3 that kind of scored it as likely pathogenic or pathogenic, which obviously would have clinical consequences if this were a real patient. Of the 48 CAMVIG users, 96% correctly classified it as a VUS and 2 as likely benign. And of the 26 other, 25 correctly classified it as a VUS. So in terms of the ACMG codes, 145 participants applied BP1 for this variant. So probably about just under half of the participants used that code. And what was quite interesting is that 114 participants applied PM2 for this case. So there's 2 really good reasons why you should not be applying PM2. One, because the guidelines say that you can only apply it if it's absent from versions 2 and 3. And we've seen it's present in every single version of Nomad and 2 cases. And secondly, as Thomas has already said, Enigma guidelines specify you should not use this for insertions, deletions, or delinces. You can see that, some people were really using this at quite high strengths. And also 67 participants applied PM4 for this variant, giving points towards pathogenicity. But again, if you're looking at that sequence conservation and that functional domain, this is not an appropriate code and is not allowed in the PM4 code. It's completely defunct and not used in the Enigma guidelines. So moving on to variant 4. This is a canonical splice variant in PALB2, C.211 plus 1 G to C. And it is completely absent from Nomad version 4. There is 1 submission on ClinVar as likely pathogenic. And you can see from all of the in silico splice prediction tools that there is a significant effect on splicing predicted. So the codes that we've used are PVS1 supporting. And the reason for this is that it actually causes an in-frame insertion, which is less than 10% of the coding sequence. It's because it's absent from controls, then the VSEP guidelines say the only strength we can use that at is PM2 supporting. And so this would give us a class 3 VUS. So looking at the results, so majority did get it right in terms of its class 3 VUS, but there's a lot of participants that scored this as a class 4 and class 5, which would have really significant implications for patients and families. Again, on the right-hand side, if you look at it kind of split by guidelines, it's kind of fairly even spread between all of the guidelines. So it probably implies there isn't like an error in the guidelines or something hard to read. It's just kind of they're very long and complicated, and it's, you know, hard to get down into all of the details sometimes. So it's probably just a kind of the understanding and experience with using guidelines. So we're going to start off looking at Splice AI, and I recommend that you do this for every splicing variant, even if it's a +1 or 2, -1, -2 canonical splice variant. Do not assume that it is definitely going to be pathogenic. Have a look at it in Splice AI. So in this case, as expected for a +1 variant, we can see that we've got a complete loss of that donor splice site that's shown kind of by that pink arrow. But in this case, you can also see that there's this increase in the donor gain of this cryptic site that's 48 base pairs away. So just before I move on, just thought I have done a whole other talk that is available on YouTube all about splicing and included kind of Splice AI and your settings and things, but just I would really recommend that you use these settings, that you have your maximum distance set at least 5,000. I know some people use 10,000, and you make sure you've ticked that reference and alternative scores. So you can see for this kind of new cryptic site, the alternative score is 0.62, which is a reasonable score for a new splice site. If you scroll down to the bottom of the Splice AI lookup that we're looking at here, and you click on this visualizations, you can then get this kind of shown graphically, which can be a little bit easier to get your head around. So you can see kind of near the end of the exon, in this case, it's going in the reverse direction. So we're going kind of exon to intron from right to left. And you can see in red is the loss of your donor site, but then you can see where the line is that gain of the donor site in gold in your into your intron. And if you count up the number of nucleotides, that is 48 base pairs. And those of you that are really good at your 3 times table will realize then that that is in frame. But actually, the PALB2 VSEP have already done all the hard work for us. And so they have this really good decision tree. And if we follow this along, we're saying it's in, we know it's like to be an in-frame splicing alteration predicted. The region of the target region is unknown. It's intronic sequence. It's going to be completely new to the gene, and it inserts less than 10% of coding sequence. And lo and behold, it's actually already said listed our variant of interest here in this box and said that we should use it at PVS1 supporting. There's a little kind of H there for some footnotes, and the footnotes kind of clearly describe what happens. So for variants targeting this exon 3 donor site, the predicted and observed outcome, which is a delta E3Q48, introduces 17 novel residues into the protein, and then that supporting strength is given based on the Provian score. And just kind of showing you it in the kind of correct orientation, from another view from Alamut, and you can see that kind of purplish arrow. Unfortunately, I haven't got a mouse, but if you can kind of see that you've got the CAG and the GTA on either side of that arrow, completely mirrors what you've got in the sequence at that exon intron junction. So yeah, that's why it's kind of given it a good score as a new splice site. So according to the Walker et al. Splicing SVI guidelines, we can also use PS1 for these splice variants. So we'll go through having a look at that. So our variant is located at the donor acceptor plus minus 1,2 dinucleotide position. So first we're going to have a look, we have applied PVS1, although it's only at supporting. And first we're going to have a look at other variants within that same donor accepted dinucleotide. If any reach pathogenic classification, we can use it supporting. If it's lightly pathogenic, we can't use them. So these are all the variants on ClinVar and how they're classified. Here are the ones that are +1 and +2, and you can see most of them are lightly pathogenic, but there's one variant that's pathogenic and lightly pathogenic with 2 stars. Actually, if you go and have a look at that record, which again, you really need to kind of read through the detail and make sure you are properly appraising the evidence, then both of those pathogenic classifications have no supporting evidence attached to them. And actually one is from 2021, so predates the VSEP guidelines. And then actually, if you look at the lightly pathogenic classifications, none of them include VSEP appropriate ACMG codes. Most of them were using much higher strengths of PVS1. And therefore, I was struggling to even get to lightly pathogenic for most of these variants. And even if we are getting to lightly pathogenic, PS1 is not applicable. Now we're going to have a look at other variants within the same donor acceptor motif, but outside that dinucleotide position. So I've highlighted a number of variants here that are kind of in that surrounding region, but you can see that they're either on ClinVar as lightly benign, uncertain, conflicting, etc. So again, there is nothing we can kind of use from that to use PS1 for. We haven't got any case control evidence available for us, so PS4 is out of the question. It is completely absent on NOMED, so we can use PM2, but maximum cap is supporting as per VSEP. Also had to look at whether we can use PM5, which can often rescue those kind of protein truncating variants, but the VSEP guidelines clearly state that for splice variants, it can only be applied for premature termination codons upstream of 1183, where PVS1 very strong RNA is applied, and that the variant must be NMD prone. So this does not apply for our variant, so we cannot use PM5. So looking at the guidelines, as I said, all participants should be using these guidelines, which the majority were, but we still have some kind of using CAMVIG or selecting Enigma, and 32 other. So 54% of participants correctly classified this variant as VUS, but 143 did classify it as lightly pathogenic or pathogenic, which obviously has more significant implications for patients. So diving into the AC and G codes used, most participants used PVS1. Actually, the majority used it at the correct strength of supporting 183, but you can see at the other end, we've got 22 applying it at strong and 73 participants applying it at very strong. I presume just looking because it's a canonical +1, just thinking that must be pathogenic. But yeah, as I've said, hopefully this is helpful so you can see exceptions to the rule and always check splicing. 38 participants also applied PP3 as well as PVS1, including one participant at very strong, but we would not do that in combination for splicing. 34 participants did apply PS1 and 23 applied it as strong, which is not an allowed strength of PS1 according to either the Walker guidelines or the VSIP guidelines. And 32 participants applied PP5. So I started doing these runs in run 3, which was back in 2019. I've just actually checked the report from that run, and we were already saying in that run, please do not use PP5. It is a disallowed code. You should use the actual evidence from those however it's been classified. And so still kind of how many years later, we still have got a number of participants still using that. And we also had 48 participants applying PM2 at inappropriate strength. So for this one, even moderate is not allowed. So, but also some were applying it strong and very strong. So my take-home message is that not all canonical splice variants are pathogenic or likely pathogenic. So please check whether it can be in frame. It can cause alternative splicing due to a cryptic splice site, or there can be rescue transcripts. And as I said, if you want a deeper dive into splicing, please check out the GenQA YouTube channel. I think that is the last one for me. I'm going to hand over to Kathleen. Thank you.
Thank you, Miranda. So I'm moving to the classification of variant 5. This was an in-frame deletion. Well, it was an in-frame event. It was a deletion insertion event in the ATM gene, but not located in a functional domain. This variant is absent from GNOMAT. In ClinVar, it has been reported as pathogenic or likely pathogenic. And there are several in silico predictions. Some of them predicted as possibly damaging, but others state that it's rather benign. So the parameters, the criteria that we have applied were the PM3 at very strong level. It's in the category for recessive disorders where it's detected in trans with a pathogenic variant. And the evidence comes from the fact that it has been reported in multiple ataxia telangiectasia families, both homozygous and in trans with known pathogenic mutations. I will come back to that later, but altogether we obtained 8 points, so we could apply it at very strong. We also applied PM2 at supporting level. This is in the category absent or rare from controls, and it's absent in GNOMAT. And the VSEP criteria are that it should be present in less than 0.001% in the GNOMAT version 4 dataset. And if it's present in a single subpopulation as a single allele, then it's sufficiently rare, and then PM2 supporting can be applied. We classified it based on that as likely pathogenic. However, there is also some older papers where segregation is demonstrated in families, and if you based on that you can apply also PP1 moderate, and then you end up with a class 5. However, these papers are not open access and they're already older, so that's why we decided that both class 4 and class 5 were correct, and there are no different clinical implications. So the results, happy to see that people are classifying the variant as either class 4 or as class 5. And the most applied guidelines are from ClinGen, and they end up with the classification as pathogenic or likely pathogenic, as we did. Which guidelines are available for ATM? So the VSAP has released guidelines that are specific for ATM. In the meantime, it's version 1.5.0, and VSAP, and Canvic UK has gene-specific guidelines. But essentially they are the same as the VSAP guidelines. However, they are still based on the guidelines on the previous version of the ATM guidelines, and they have some additional points of specification. But for this variant, I could not find any different interpretation. So we applied the PM3 very strong. You can apply it when it's reported in AT families, and both in homozygous and in compound heterozygous states, with another pathogenic variant on the other allele. But the phenotype of ataxia telangiectasia is something very specific, and there is generally not a differential diagnosis. So when you find a publication stating that a clinical diagnosis of AT is granted, then you can use it as a confident diagnosis. And this is important to apply the criteria, because for a confident phenotype, then you also it must also include a laboratory result, and I will come back in the next slide what's the difference between a phenotype and a laboratory result. So for a confident phenotype, you need at least 2 laboratory results, and it's from result 1 to 4, or the presence of a clinical feature from 1A, 1B, and the presence of a laboratory result, or the presence of a clinical feature 2 or 3, and a laboratory result. And then for a consistent phenotype, there are less strict criteria, so they're not so restrictive. It's the presence of 2 or more clinical features of ataxia, or the presence of 1 clinical feature and 1A or 1B, and either clinical feature 2 or 3. This is not understandable except if you take it together with this explanation, where there is in detail listed which clinical features should be present. So it's going from cerebellar ataxia, oculocutaneous telangiectasia, and immunodeficiency. And then what do we mean with laboratory results? Well, you need to determine the level of protein present, and this is often done by Western blotting, and this should be present at less than 15% compared to controls in the patient you're investigating. However, sometimes this level is higher than this 15%, and in that case you need to show that the kinase activity is either absent or negative, or very low, or only marginal or residual. In case the protein levels are higher than 50%, this occurs sometimes in patients with a missense variant, a leaky splice variant, then the variant should result in a kinase dead protein. So the kinase activity should also be investigated. For our variant that we are here discussing, there are 2 papers available. However, they are a bit contradictory. In the paper already published in 2000, the variant was from Becker-Catania et al. The variant was described that the variant was associated with complete absence of ATM protein in one patient who is homozygous for this variant. And then in the 2012 paper from Verhagen et al., the ATM protein was still detectable, but there was no ATM kinase activity in 4 patients that were homozygous for this variant. Other laboratory results that are can be easily determined is the serum alpha fetoprotein that needs to be higher than 65 micrograms per liter, but it should be in patients older than 2 years old, because when they are younger, this level can be higher, and which can be perfectly normal. The fact that AT patients are sensitive to ionizing radiation, and that can be determined in fibroblasts or lymphoblastoid cell lines, is also criterion for AT patients, but it's not ATM specific. There can also be other DNA repair disorders that show sensitivity to ionizing radiation. So it's a less strong argument than really looking at the protein level and the kinase activity. And then in some patients, there is a presence of a specific chromosomal translocation in the peripheral blood cells, and it should be present in more than 5% of the cells. So based on these laboratory and phenotypic results, you can give points, and as we have here several patients that were homozygous for the variant, we can give these 4 points altogether, because you can include maximum 2 homozygous patients, and then variant confirmed in trans with another pathogenic variant and a confident phenotype. Per patient you get 4 points, so we easily, based on the literature that's available, we easily come to 8 points, so a very strong argument. Another argument that can be applied is the PP1. So it's the co-segregation with the disease in multiple families. It should be clear that the affected relatives must have both variants identified in a proband. This segregation argument cannot be used for the autosomal dominant condition. So having the variant present in patients with breast cancer cannot be used as under PP1. And then the strength of the criterion is based on the number of affected relatives, and we have been careful, and we have seen it in different papers. In the paper from Van Belzen et al., it was a brother and sister who were homozygous. And in the paper from Verhagen et al., it was observed in 9 patients from 7 families. Not completely clear if they were really independent, but we had at least 2 affected relatives, and that's why we have chosen to apply it at the moderate level. So, which guidelines were used by the participants? So the VSEP guidelines in the ATM guidelines that should be used were applied by a quite limited number, 43 of the 312 participants, and there were several who combined the ACMG 2015 guidelines with ECGS guidelines, and all other combinations. So, of course, there were also many people who applied the CANVIC guidelines, which should be similar as the VSEP guidelines. Results, so the large majority classified the variant as either class 4 or class 5. There were a few that reached class 3, a few that reached class 2, and there were also 6 persons who indicated that they could not classify the variant. So we hope that with these with this extra information, this will the skills will improve to classify variants in ATM. Unfortunately, there were also some ACMG codes erroneously applied. Some applied the PS3, it's for the functional assays. However, it's clearly stated in the guidelines that you should not use phenotypic evidence, so the lack of ATM activity in cells from a patient with AT as functional data. You should consider this as part of PM3. PP5 was also applied by some of the applicants. So this Miranda, as Miranda indicated this before, this should not be done, and this is already known since a long time. And then the other codes that were erroneously applied, the prediction programs. So for the ATM guidelines, you can only use for Splice AI, it's for splice variants, or the Revel tool. And however, Revel is not suitable to evaluate indels. For your interest, to obtain PP3, you should have a missense variance that reaches 0.7333. PVS1 is only relevant for truncating or splice variants. As this is an in-frame insertion deletion, this should not be applied. And then PS1 was also applied, so the same which means the same amino acid as previously established pathogenic variant, regardless of the nucleotide change. This can only be applied for missense or splice variants. And then the last one that has been applied by some of the people that were registered for this scheme is that the variant is more prevalent in cases compared to controls. And you can apply it at a strong level in case the p-value is smaller than 0.05, and the odds ratio or the hazard ratio or relative risk should be higher than or the same as 2. And then I calculated it for the Bridges study based on data I could find in LOVD, and there I obtained an odds ratio that was well below the cutoff of 2. And it should be clear that you should not use data from publications on that are a bit biased towards patients with cancer to determine these odds ratio, and then compare these prevalences with prevalences in GNOMAD. Then let's move to variant 6. It's the last one. It was a variant in RED51D, and it was the loss of the start codon. And in GNOMAD, this is not present in version 2.1.1. However, it was indicated that there were, there are 5 start losses in GNOMAD 4.1, but not in the South Asians, and it's a different one than this one. In the UK Biobank, there is 1 in 480,038 females. And in ClinVar, there are conflicting classifications going from pathogenic to unlikely pathogenic and uncertain significance. The in silico prediction splice AI does not predict aberrant splicing, and then some of the other tools like Max and Scan indicates that the splice site is the score is increased. PVS1 is what the criteria that we applied was PVS1 at the supporting level, because there is an in-frame methionine 16 amino acid positions downstream. And we also applied PM2 at the supporting level because it's absent from GNOMAD, but present in one out of more than 480,000 females in female alleles in UK Biobank. So we ended up with a class 3. And this was also the classification that was obtained by the majority of the applicants and participants, and yeah, most people applied the CANVIC guidelines. What guidelines are available? Well, I can keep it brief. For VSEP, there are no RAD51D specific guidelines. So it's logical that the majority of the participants applied the CANVIC UK gene-specific guidance. However, if you go in detail, then it's they apply actually the same criteria as for the ATM variants. So they refer to these. PVS1, we used it at the moderate level according to the Tayoun et al. Guidance. And you can apply it because there is if the initiation the initiation codon, and there is not a known alternative start codon in other transcripts, which is the case here. But then you have there are two options. Either there's one or more pathogenic variant upstream of the closest potential in-frame start codon, which is at methionine 16 at amino acid position 16, and then you can apply it at PVS1 moderate. However, to our knowledge, there is no pathogenic variant upstream of the closest potential in-frame start codon, and then it's PVS1 supporting. And there has been a nice publication. It's already published in 2011, where they have indicated that there is an in-frame methionine at amino acid position 16, as here indicated with the pink arrow. However, it's predicted to be a relatively weak translation initiation site, and removing part of this amino terminal domain of the RAD51D is thought to have a functional impact because it plays an important role during homologous recombination. However, we did not find any more recent literature about this. And then PM2 supporting, the variant A to T is not present in GNOMAD version 4.1. In the GNOMAD there are 5 start loss alleles, but it's A to G, and it's exclusively present in the South Asian population, so it looks like a founder effect there. For the rules for the CANVIC UK are to use the female-only population controls, and VSEP is working on guidelines for RAD51D, and CANVIC has already had contact with VSEP, and at the end of July they have responded how to deal with PM2, and it's a bit similar as for ATM. So if the group max FAF is smaller than 0.001%, you can use it at supporting level, and it's if the group max is not calculated, because specifically if there are only low numbers, you can use it. It's the same cutoff if there are, regardless of the number of alleles present in GNOMAD, but if there is if it's higher than this cutoff, then but there is only one allele in one population, you can still apply PM2 at supporting level. And these are the similar cutoffs as applied by VSEP, the ATM, in the ATM VSEP guidelines. Some participants applied some codes erroneously. Some of you applied PS1 at strong or moderate level. This is about the same amino acid change as previously established pathogenic variant. However, at the moment, there is no other start loss classified as pathogenic. Some referred to ClinVar, but if I looked in detail at it, there were conflicting interpretations. So you cannot consider it that there is already clearly established that start of the loss of the start codon is a pathogenic variant. PP5, same as said before, you should not use. And PS4 moderate was used as strong, moderate, or supporting. So it's again the prevalence in cases versus controls. And also here the arguments that were applied were really based on odds ratios calculated using HBOC populations versus GNOMAD the GNOMAD as controls, and this is of course too much bias. And I think this is the last one for me, and I would like to hand over the slides, the talk to one of the organizers.
Thank you, Kathleen, very much indeed. It's back to me. So thank you also, Thomas and Miranda, for a fantastic amount of information, huge amount of detail there. And we're having great response from all of the participants. We've got lots of very interesting questions to come to. So just before we kick off with the Q&A, I'd just like to summarise some of the findings then from the run as a whole. Oh, if we just go back, please. So we can look at our bubble charts. We always love our bubble charts. So the larger the bubble is the number of participants that declared that was the classification for each variant. The variants are along the x-axis, so you can see 1 to 6. So anything that's a red bubble is not the expected classification, whereas a green bubble is the accepted classification. So I think, again, you can see a variability. You can still see some very large red bubbles where that would be the incorrect classification according to our expert review. But then again, on for example, the variant 3, here you can see that either a VUS or likely benign was the majority of the participants' outcome, and the very, very small outliers were a class 5, 4 or a class 1. So I think, again, it's very dependent on the variant and the gene and what guidance is available. So I think we can, again, see the spread of the classes there. So next slide, please. Lovely. So this is a summary of both of the EQA runs in 2025, the list of the different variants, the genes covered and the classification, and again, the variability be depending on the guidelines. We're in the region of 400 plus registrants and then participants with more than 800 registering over the whole year and over 600 actually participating. I think what we see annually is definitely an increased participation in the first run in the annual cycle. So no surprises for 2025 there. Also, we have a series of these 4 webinars supported by AstraZeneca and MSD, to which we would like to thank for the educational value. They are all, and webinar 4 will also be joining the other 3 available free of charge on the GenQA and EMQN YouTube channel. So please do pass that information on to any colleagues who would be interested to view them. Next slide, please. So a huge thank you to our expert panels. We rely on them incredibly to give us the expert advice and also their knowledge of the guidelines, and a lot of them are very heavily involved in writing or ratifying current guidelines. So a huge thank you. And also to Fergus Couch, who was from the Mayo Clinic in Rochester in the States, for his incredibly educational webinar earlier in 2025, which again is available on the YouTube channel. Next slide, please. The summary report for the run that we've just been discussing today will be available towards the end of December or into early January, so please look out for that. You'll be notified when it's available. We do plan to run a further assessment in Run 16. We may need to charge. We're in discussions just now about educational grants going forward. So please be on the lookout, and we will inform you as soon as possible when you can register through both EQA providers. Next slide, please. Right, Simon, over to you to go through the raft of incredibly interesting questions that we have available. We've got 10-15 minutes to see what we can cover. I'm aware a lot of them actually are very in-depth with expert knowledge required and maybe a bit of digging through the literature. So we'll probably follow up a lot of them in the EQA summary report, but let's have a look at what's available for us today.
Yeah, thank you, Sandy. And just to concur on what you just said, I think the amount of feedback we've had for this webinar is far in excess, I think, of what we've had in any of the previous webinars. So thank you to you, the participants, for contributing to that, because obviously this is your webinar. We are trying to make sure we can answer your questions, but you are right. Most of it's very detailed, which shows a real high level of engagement in terms of the actual QA itself. I'm going to start off really addressing variants 1 and 2. So Thomas, these are coming your way, I'm afraid. Line yourself up.
Yeah.
Hopefully nothing too difficult. We have triage. We've been through a lot. We've had 40 questions so far, which is brilliant to see. So there are a couple of questions relating to variant 1. So why did we not apply PP3 supporting to the classification of that variant? There are two individuals asking the same question.
Yeah. Okay. As I understand it, and I think it's also written in at least in the Enigma guidelines, the PP3, you can use it for missense variants or indel in-frame deletions or insertions. And Miranda and Kathleen, you must correct me if I'm wrong. And also for splicing. And I mean, in this case, it's a frameshift variant that leads to a stop after the original stop, and I'm not even sure that you can get a base del score for variants like that. I mean, I have not tried it, so I guess you can't. So I guess that would be my answer. That's why you cannot use it for this variant.
Miranda, Kathleen, do you have anything more to add to that at all? No. Okay.
Only that agree with Thomas. I don't know if the participants thinking because it's in frame, but yeah, you're not going to get a Basedel answer from that with that kind of insertion of novel amino acids.
I think that would concur with what one of the questioners had actually said, Miranda. So I think that's probably the answer to that. Okie dokie. Another question for variant 1, Thomas. So this relates to PM2, but I think also there was a number of comments actually just more general about the classification of this variant and variant 2, in fact. So actually what I'm going to do is I'm going to come to that first, because I think this is quite an insightful question. So several labs have reported a variant at the same location, and this is for variant 2, I think, in fact, actually. Sorry, I'm just reading through it. It's quite a detailed question. I want to sort of summarize it in a way that's understandable. Given that the variant, I think it's in case 2. Let me just confirm it. Let me just go back to it 1 second. So yes, here we go. So variant 1 bracket, we learned that now that PM2 cannot be applied for an insertion deletion or an del ins. But for variant 2, apparently the application of PM2 is applicable, even though there's also a deletion and the gene-specific guidelines state do not apply insertion, deletion, or del ins variants. The questioner is asking, why is this the case?
Yeah. I think it's a good question because we have actually discussed this a lot, and actually it's also rather confusing that in one set of guidelines you make this comment regarding the use of PM2, and in other guidelines it's not even mentioned. So there is a difference there, and as we discussed earlier today in the expert group, I mean we think this is based on rather old data. Also where maybe the quality of the sequencing data were in the population data were weaker, and we actually think that the bigger the deletions or insertions are, the better these techniques are able to be correct. So I mean, for the small in deletion insertions, it has been a problem in the past that is the data reliable, especially I guess in repetitive sequences. So I mean that would be my question to my answer to this question. So yeah.
So
I think it would be changed. I'm pretty sure Miranda has, because she also mentioned a paper and has some insights into that.
I was going to come on to that, Miranda. Did you want to add to what was said as well?
The sound went funny, but I think, can you hear me okay?
Yeah.
Yeah, good. So yeah, it's in, if anyone wants a kind of deeper dive into it, as I did earlier, it's in Appendix G of the Enigma guidelines, but the paper that it references, I can maybe stick it in the chat after this, we'll definitely put it into the final scheme report, but the paper that it references is from 2021, so it's kind of pre-NOMAD version 3 and NOMAD version 4, and yeah, as Thomas said, they had this kind of problem that there was a kind of high error rate with some of those kind of homopolymers, but actually they found for deletions that were greater than 50 base pairs, it was actually more accurate, which is kind of now sequencing technology has kind of moved on a lot. The bioinformatics has moved on a lot. We've got NOMAD 3 and 4, which I've already said has got much more stringent QC checking. So personally, I think the Enigma guidelines probably need to kind of review that, but we have obviously stuck with the Enigma guidelines, and that's why we have applied it for the CMV, but not applied it for Enigma, but it's different for CAMVIG. I appreciate for participants, this is difficult and confusing, and everything's always in flux and gets updated, but yeah, that's where we're at.
Thank you both for your answer on that. I mean, I think there was a lot of questions generated for that. It's clearly been a challenging question, well, 2 variants for the participants, but we will, as you stated, make sure that we update the report and have a general comment in the report that covers that area. Sandy, I'm going to hand over to you for variants 3 and 4.
Lovely, thank you. So the first comment is actually related to variants 2 as well as variant 3, and it's why is BP3 not applied for both of these variants? Is it to do with the repetitive regions, and are there definitions of how repetitive and how conserved these regions should be for that criteria? So Thomas, do you want to refer to that?
Yeah, I can start. Yeah, I mean, it's stated in the original guidelines for ACMG in 2015, and when you look at the gene-specific guidelines, it's stated that this criteria is not applicable. So therefore, you cannot use it, and I think it's mainly due to that some of these things are also incorporated in the bioinformatic prediction in some of the other criteria. So I guess that's the main reason that this has been skipped, and I don't remember in other schemes. I mean, for other gene-specific guidelines, is it the same? Has they also skipped this as non-applicable? I don't remember actually, but for BRCA1 and BRCA2, it's not applicable.
Okay, thank you. Miranda, do you want to come in if there's any change of view for variant 3 in particular?
It's one of the kind of bugbears of the kind of ACMG 2015s. Quite a few codes are a bit subjective, and so there isn't any kind of hard and fast rule as to what you would say is repetitive. But I think I showed the sequence around there. You'd always go and have a little look at it and see, you know, sometimes you can see that it's clearly a repeat, and that clearly is going to kind of, we know that there's kind of, yeah, tandem repeats and repeat expansion disorders and things. So something like that, you would definitely use BP3 in that circumstance, but not in, certainly not whole exon deletion, and not in the example in variant 3 either.
Great, thank you both very much. So Miranda, just sticking with you, another question about variant 3 and one of the criteria. So why not apply BS1 supporting with the ClinGen guideline?
So this was the BRCA2 in-frame one. So we had three alleles in Nomad version 2, and then 2 alleles in versions 3 and 4, which is below the kind of cutoff for BS1 supporting, but not allowed. You therefore can't apply PM2. It's in that grey zone. We always have to have a kind of a buffer that, you know, some things will meet too common, other things will meet very rare, and yeah, you have something in the middle, which is this one.
Yeah. Which is where subjective evidence and your professional judgment has to come in, isn't it? Yeah. Thank you. Right.
I think it's actually very important that you have those zones, for a lot of the codes.
Yeah. Right. So we're going to move on to version 4. Now, there's been a lot of queries coming in about this PALB2 variant, particularly around the sort of expression side and functional studies, so about the RNA data. So it's a couple of people have come back and said that there's RNA data is available, particularly according to the splicing table. PS1 strong can be applied. And also there's RNA data available, which is not indicated in the ClinGen decision tree, which supports PVS1 supporting. Do you want to comment?
Yes. So when I showed the PALB2 VSEP PVS1 decision tree, I didn't highlight it, but one of the, I think it's the G to A variant, was in a slightly different color. And that's because there is RNA splicing data available, which supported that insertion of those 17 amino acids and confirmed that it was in frame. And that was why it's been given the strength of PVS1 supporting. I'm not aware of splicing data for this variant, the G to C, but the Splice AI is exactly the same for kind of all of those variants. It's got the same predicted effect, so that's why it's applying PVS1 supporting for all of them.
Right, that makes sense, and that's why it's on ClinVar for either likely pathogenic or pathogenic for the G to A variant.
I think it's the G to T. Is it the G to T variant?
T. Yes,
G
it is.
T variant is the one that I highlighted that is on there 8 times. 6 is slightly pathogenic, 2 is pathogenic. But if you look at the 2 pathogenic, 1 has nothing at all, just completely blank, and effector status is unknown. And the other one is from 2021, so pre-VSEP guidelines, and actually looking at the likely pathogenic ones, they're not using kind of PVS1 supporting. They're using either not giving the codes or they're kind of, so again, lots of them are pre-VSEP, and obviously not. I think it's very easy to see a +1 variant and go, that's pathogenic, and apply PVS1's very, very strong, which is one of the reasons we include variants like this in the scheme to help kind of raise awareness that they're not all pathogenic.
And also a good example of not just to take what pops out of ClinVar, etc., as written, that to actually go and have a look, and because things evolve, and that's really, really important.
Yes, 100%. There's a lot of ClinVar is very good. There's a lot of data in there, but always contact. If you're going to use that evidence, and particularly if that evidence is going to change your classification, contact the lab. Most of them are really, really good at getting back to you really quickly and kind of giving you the evidence, and then you can critically appraise that and put that evidence into your variant classification rather than the PP5.
Brilliant, thank you. There's another question about talking about looking at the compound variant. I just need to work out which actual.
I think that'll be the ATM one.
Yeah, it's the ATM one. So we'll maybe move on. Kathleen, will I do this one, Simon? Because I'm quite interested.
Go for it, Sandy. If you're up for it, you go for it. Yeah,
Thanks, Miranda. Kathleen, right, so the ATM variant, the compound variant, okay, so it's not listed as a compound variant in Nomad, but both changes, if you take them separately, are present. But then the individual submitting this question saying inspecting the reads indicate that they are actually linked. Therefore, in their understanding, PM2 cannot be applied. What's your thoughts?
Yeah, to be honest, I did not go into detail in GNOMAD checking if these individual variants, because of course it's not so easy to find out if they are in cis or in trans. If you get you have to go very in much detail, so I'm not sure how frequent they are then. So on the spot here I cannot answer if they reach the cutoff or not. But it's very nice that people go in such detail for this exercise, and that they really try to find out in GNOMAD if the variants, these more complex variants, are really absent, and that's also demonstrating what we discussed before, the PM2 interpretation. It looks straightforward, but it can be way more complex. Yeah.
And also a good example of sort of stepping back and seeing, is it in cis or trans, and how that could maybe change classification. Are you looking at two variants, or is it a one event? And it's very complex.
Indeed, fully agree. Yeah, with that.
Thank you, Kathleen. Simon, back to you then, please.
Thanks, Sandy. I think we've got, given the time, we've got time for one more question for you, Kathleen, on variant 5. So for variant 5, can we use BS3, as there is functional evidence in publications showing no damaging effect on protein function, as there is an alternative initiation site at methionine 10, I think it is. So they've given a couple of papers to justify that. What do you think about that?
And it's for the ATM variant, or?
The variant 5, I think.
Variant 5 is the
BS3.
ATM variant.
ATM.
Yeah.
Because methionine 10 is. Maybe I did not fully understand the question.
It's just saying that we can we use BS3, as there is functional evidence in publications showing no damaging effect on protein function, because there's an alternative initiation site at methionine 10.
Okay, in that sense. The only literature that I found was really about Western blots that have evaluated the presence of the protein, and there, as I indicated, it was a bit contradictive, and some found presence of protein in homozygous patients and others did not find it. But the ones that what was clear and what was consistent, at least amongst the papers that I have been reading, was the reduced ATM kinase activity. But as I said before, we cannot put it in the functional assays here in the VSAP. According to the VSAP ATM guidelines, you have to use it as evidence in the PM3, specific for the phenotype and related to the recessive disorder. So it's a bit confusing, and to be honest, I was also first, when I did the quick classification, I had also applied the functional assays, but then when reading in more detail, I saw that it's a bit for me a bit contra-intuitive that you should use then put it in the PM3 evidence.
And this goes to show why the educational side is important, isn't it? Because we're all learning constantly as a consequence of this process.
Yeah, indeed.
Thank you, Kathleen, so much. Right, I think we are probably out of time, Sandy. It's just gone past our allotted time, and we are aware that obviously that a lot of the participants are hard-working individuals in the laboratory. We don't want to take up too much more of your time, but we will, as we said before, make sure we haven't addressed every question, but we've addressed most of them. But we'll make sure that we pick all of this up where we haven't addressed it offline in the summary scheme report, or directly respond to you where we have an email to do so. I think that what we'll do is we'll draw the webinar to a conclusion. I wanted to pass on my thanks to you, the participants, for dialing in. We hope this has been helpful for you. Lots of engagement, so I'm pretty sure it has been. Do, as Sandy said before, check the EMKN and GenKA websites for a copy of the webinar when we publish it in a couple of weeks' time. And the reports will actually be out end of December, beginning of early January. Thank you to the organisers as well, and the support from AZ and MSD to be able to provide you with these educational webinars. I think everybody can see the value in it. Very helpful and really good for the educational component. A big thank you to our speakers. Thank you all three of you. Brilliant job as always. It's very difficult and it's really hard to be put on the spot with these detailed questions, but I think we can see that the engagement from our participants is brilliant. And finally, a big thank you to my co-chair, Sandy Deans, for her support as well and for doing a great job of co-chairing. So wishing you all a good afternoon, and thank you once again for dialing in. We'll speak to you hopefully next year in Run 16.